So the idea of biocontrol is we are going to go back to these foreign countries and look for natural enemies to reunite with their host. We have two things basically. What we're trying to do is we're going to reduce the density and competitive ability of these plants, one, through direct actual action of the insects feeding on the plants and destroying them, or through this indirect method over there where we have a thistle head, here, and there's a little fly larva inside there, and it's created this gall. It kind of acts like a cancerous tumor, so to speak, to the plant, and it acts as a nutrient sink. And so those are kind of the ways biocontrol works. Now, where does it work? In Oregon, most of our stuff is rangelands and pasture oriented. Gorse sort of fits in here, also somewhat in riparian. And yes, we are seeing gorse on the Oregon coast in these riparian zones, in these wetlands, just above the water level, and we're seeing the same sort of thing. So that's a problem. And then over here in natural areas, this is probably the big problem where we're worried about gorse, would be natural areas and pastures, and somewhat forests. So it just kind of gives you an idea of where we're doing things. Now, to get a biocontrol agent into the United States, it takes about 3 to 5 years or more. It may cost $250,000 to $500,000 because we got to make sure that when we bring something in, it isn't going to be a pest. So the first thing we do is we identify where the weed is from, where is it native. Then we go over there and we do what's called foreign exploration, where we look for the weed which is a wildflower in its native habitat. We go in there with scientists and we look for insects that are natural enemies that are feeding on them. And then once we do that, then we test them for safety. We want to make sure that they're not going to become a pest when they control, or if they control, their targeted weed here in our environment. Then we go through a NEPA documentation process, because if we're going to release them on federal lands, and they may have some potential impact, we have to go through that. Then we import the critters. Usually they're insects, so most biocontrol people are going to be entomologists. And then we import them, and then we make a release in the new environment. When their populations build up, then we go into the redistribution phase. Now, so what we try to do is when we're going to work on a particular plant, one of the things we look at is we look at the whole plant. And we try to find natural enemies that will have some impact somewhere on this plant, and that it will be meaningful. So you can see this is Dalmatian toadflax. There's quite a series here of insects that feed throughout the plant. Usually what we try to look at and target is we try to target the reproductive part of the plant, and then we try to target the photosynthetic part of the plant, and then the root storage area of the plants. And we try to find natural enemies that will attack those. Do we always find them, and are they always good? The answer is no, but we try. Okay, there are benefits of using biological control. First, there's no harmful residues in the environment here, unless you're going down the street in a motorcycle and one hits you in the face. They're host specific to the target weed. They maintain themselves. It's long-term control. Once you release them, they pretty much do it. They synchronize themselves with the development of the target plant. They're economical on low-value lands where, you know, the spraying and efforts that you put into some weed control projects cost way more than what you get on the return of those lands. They can search the area and find the weeds where you can't find them. They can smell them and fly right to them or crawl to them. And there's pretty low resistance to the particular targets, you know, versus pathogens. Okay, now there are some problems. I'm not here to tell you that biocontrol is going to be a panacea. Generally, initially, it's hard to get them because a lot of the plants that we're working on and where these insects and things are in areas where the governments don't like us to be there. So we have to work with scientists from different countries and kind of go do an end game around that. The degree of control is usually density dependent, and it takes a lot of time. A good project like on tansy ragwort that we've done here in Oregon, it took 20 years from the time those things were released until we started getting regional control. With gorse, it's going to take a lot longer. We have to match the biotypes if it's necessary. We need to worry about host specificity once they're released here, because you can't test every species that you have in a new environment against these insects before you release them. And so in some cases, rare in cases, there may be some impacts from the biocontrol agents on plants that you don't want them to feed on. And then some places the control isn't desired. You know, beekeepers like yellow star thistle and things like that, and cattlemen don't. And then there's a problem down here where it's hard to get out there once. It's a project like Gorse it was started back in the 50s, and then nothing happened for a long time until the 90s, and now we're working on new insects that I'll talk about. So once we get biocontrol agents established, and if something else needs to happen, you can't just go out, release them, and then walk away. So what we do is we monitor, and this is where we try to get people like you involved when we're doing new biocontrol projects. We want to first find out, are they established? Then once the population builds up, we collect and take the surplus, and then we spread them to new areas, or we watch them do that on their own. We want to determine the attack rate. If you're working on gorse, how many pods have weevils, seed weevils in them? We want to know what the damage level is, okay? How many seeds in a gorse pod were attacked and the seeds killed? Then we want to know, where do they occur? Okay, we're going to work on gorse pretty much from Astoria to Brookings. We want to know, where do the biocontrol agents occur and what kind of numbers are they? And do they have any impact on the host? And if they do, how long does that last? And then the real important thing, which we all want, we want to measure changes in the plant community. Did they do any good? I mean, just getting rid of the weed is one thing, but you know, is there a change in the plant community? And we have to document that just to make sure. Okay, with biocontrol in Oregon, we have about, as we said, it's about 75 species that have been introduced, mostly insects, beetles, flies, and moths, 3 mites, and here's the gorse spider mite right there, so that's kind of a cool thing. A nematode and 2 pathogens, one on rust skeleton weed and the other one on yellow star thistle. So, but you can see pretty much entomologists are the guys who usually do biocontrol. So on gorse, the biocontrol on gorse started, I think, in the 1950s, and one of the problems that we have in working with gorse is it's a plant of many habitats. Most of them are coastal. Some of them are so close to the ocean, we've got these steep cliffs and stuff, it's windy, it's cold. In areas like this, probably biocontrol is not going to be an option. It's just too harsh and it's too difficult of a system for these insects to be in and to do well. So if you've got these salt spray cliff faces infested with gorse, you're probably either going to have to go out there and mechanically remove it. I don't even know if you can spray it in a place like that, it's dangerous, or you just have to learn to live with it in those kinds of areas. And sometimes in some years we see this stuff gets frosted pretty bad, but it always seems to come back. Okay, but in the areas where you can do biocontrol of gorse, and for most of my work, it's anything usually inland more than a mile or so inland. We have a suite of different things that we're finding working on gorse. There's the gorse seed weevil up here. There's the gorse spider mite. And then we have this accidental adventive moth here that's a defoliator that feeds on the soft shoot tips when it's growing in the spring, but it's not technically a biocontrol agent, it's just an adventive natural enemy, but you guys will see it. Okay, gorse doesn't have a lot of redeeming qualities. One of them that I've noticed is after you get over the dunes and past the deflation zones and whatnot, at the what I would call the eastern edge of the gorse, usually in those areas, you get a lot of hummingbird nests. They really like gorse. And so if you go along these, you'll find, I found like, what, 4 hummingbird nests in about 20 yards walking through the gorse. And where the spider mites were abundant, they really liked that because they could take the webbing from the spider mites and get lichens, and so you see lots of those. What are some of the other things we've seen where gorse has been used for? I've seen it carved up and made pencils out of gorse wood. I think that's about it, other than maybe some spectacular pictures of where people thought it should look like the Scottish coastline. I've seen that on a few postcards. but I don't think of a whole lot of other redeeming qualities about gorse. You know, if somebody asks you, well, how do you tell gorse from Scotch broom? I always just say, shake hands with it, recognition and release are simultaneous. Okay. As far as biocontrol, the first thing that we started out with was the gorse seed weevil, and they're pretty small little guys. I think I've got a couple here, and I'll pass them around. There's a jar here with some gorse mites, and then there's another one in here with some weevils. I'll just pass these around. You probably won't be able to see them here in the dark, but they'll still be up here, and I've got a bag full of some gorse here with some mites on it. We can take a look at that later, maybe even make a release later this afternoon. Okay, so anyway, the gorse seed weevil has just one generation a year. The adults are pretty much, you can just about find them almost throughout the year, but they're abundant right out there. They overwinter as adults, and then in the spring, when the gorse is blooming, you can see the feeding marks right here. Here's the weevil right here, and another one. Then as the pods elongate, they lay their eggs into the pods, and then the larvae actually feed on the seeds. And if you open the pods, about the time the pods start to turn black, about before they're ready to dehiss or while they're doing that, if you open them up, you can actually see in there, you'll see where there should be a seed, there'll be a weevil there. I don't remember if I have a picture of that or not. Oops, I don't. Dang. I don't know what happened to that. Oh, okay, I know why I don't. Because I don't have an, it was a slide that hadn't been scanned yet. So when I made that transition from slides to PowerPoint electronic pictures, I left it behind. So anyway, the gorse seed weevil is a pretty good critter to have. They do a lot of damage to the gorse seed production, but I haven't seen very many sites where it gets much above 60%. Sometimes it can be 20. In some little dry areas where there's not a lot of gorse, I've seen them get as high as 90%. But from what I've seen in the literature and working with Scotch broom and other plants, the seed production on gorse has to be reduced at least by 95% or more for this to be an effective agent. So this insect, it's pretty much everywhere there's gorse, but is it doing anything to the plants? Well, the adults will feed on and pit the plants here on the edges, and on some of these spines you'll see little brown spots or whitish spots on those. That's from the adults feeding, and they really have no impact on the plant themselves. In fact, it just doesn't. So what do the gorse seed weevils do? They pretty much slow down the spread so there's not as many seeds going everywhere. Okay? Now that brings us to the gorse spider mite, which we released in Oregon, trying to remember what year, somewhere between 92 and 95. Years now are flying by too fast. But these are the gorse spider mites. This is an electron microscopic photograph, but this is what they look like if you look at them under hand lens or a microscope, just like a dissecting scope. But this is what you'll see. When you look for gorse spider mites, you're going to look for the webbing, and you're going to see this kind of a red velvety look on the gorse stems. They particularly like the old growth. They don't like the new soft growth. That's just not what they like. They like to feed on the old hard stems. They are sucking organisms. They have these fangs. They stick it into the cuticle of the plant, and they suck out the juices. That's what they do. And when they do that, they create kind of a bleached appearance. I'll show you here in a minute. So basically, this is what you look for. And gorse mites, they move slowly through the stand, and you could kind of consider them as a biological fire moving very slowly through the plants. They like it where it's warm. They like it where it's not too windy. And so that's where you look. So if you look on the south side of the plants, that's usually where you'll see them. When the wind is blowing, like here from the west all the time, they're going to be going from the west to the east. And you have to take them again and bring them back and start them again, which we haven't done recently, but we'll talk a minute in a little bit why. So what we used to do after we got the gorse mites going and we had surplus, we would go in sometimes with chainsaw gangs and go through a big patch of gorse, and we'd get all of these little colonies of mites, throw them in a bucket, And for a couple of years, we drove throughout the coastal area, especially around here in Bandon, and we'd reach in, we'd be driving along, roll down the window, and the person in the passenger side would clip a piece of gorse about that long with some mites and then just fling it out into the gorse. And we just went through the whole area playing Johnny Appleseed with gorse mites. And it worked pretty well. By 1990, I was either 95 or 97, here in the Bandon area, if you saw gorse, you saw gorse spider mites. And it's incredible what happens when their populations build up. Here you can see, this is all gorse that's been bleached, and you can see just the webbing is just incredible. Usually you see right at the beginning of the colony, you'll see these really dark, the red, velvety red mites, and there's just, you know, probably millions of them right there. And then sometimes they get out on the edges of the stems and there's nowhere to go because there's all of this stuff behind them. They form these mite sickles and they just drip right off of there, and here they've hit the end of the gorse. And they're just out here on the sand and they just sort of go off looking for more gorse, and they web everything up as they leave. They'll cover everything with webbing. Rhododendrons, roses, bicycles, the lawn. In fact, during that outbreak period of time, there were people complaining here in Bandon that these gorse mites were going to eat Bandon alive. And that became a problem later for a short time. So anyway, once these things get going, you just saw gorse, you just saw webbing. It looked like arachnophobia too. Everything was webbed, and it was really spectacular. And this happened, and so we were thinking, this is so cool. We finally have something that's going to do something to gorse. And then, well, okay, so I haven't hit to that part yet. I will in a second. So this is up at Baker Beach on Forest Service land. This is a plant right here that got hit heavy during that previous slide, and this one had not been attacked. So this is 1 year post. There's a Scotch broom plant right here I want you to look at. So you can see that the heavy mite attack from the previous summer reduced the flowering by probably 95%. So there's very few flowers here, and compared to this plant that wasn't attacked. And so we thought, wow, this is really cool. We're going to actually do something on gorse. Didn't we, Glenn? Weren't we excited?
We were excited, yes.
Yeah, come on, use your excited voice.
We were excited.
I told you. Only I wasn't that excited. Just Glenn. So anyway, so we thought that'd be, oops, really cool. Let's go back one. So we went back the next summer after that bloom, and this plant had all been hammered, and this one got pretty well hit. Here's our Scotch broom plant there again. And that was during the arachnophobia, so that was 1997. And then we went back in 98, and this plant had recovered. Here's that Scotch broom plant, so would this one. This one probably about 20%, 25% bloom. So we were starting to see this amazing impact depending on what time of the summer the gorse mites hit the plant. If they hit it early, during the early growth and whatnot, then it didn't produce a lot of flowers. If they hit it too late, the flower buds had already been produced, and it could produce more flowers. They don't, but because they like the old growth, they don't go back onto a plant that they've hit as readily. And so you have to wait for this. So you kind of get this sporadic business of here they are, and then they're over here, and then they just kind of have to move around. And so it wasn't this massive webbing of the plants year after year. So it kind of became sporadic. So it didn't look as cool as what we had expected. So this we thought would really make a difference, and then we found out otherwise. Enter the villains. We just thought everything was going to be really cool. And then in 1998 we went out to a lot of our sites, and we found that the gorse mites were all but disappearing, and we couldn't figure out what was going on. We talked with Dr. Croft at Oregon State University. He said, well, you might be concerned that there are some predatory mites that like spider mites that they use for biocontrol agents. So pest mites in greenhouses and in fields and stuff. But you should be okay because most of these pests, or these predatory mites, don't exist much further north than Redding, California. That's what we were told. Well, it didn't work out that way. What happened is we found that the mite colonies here in the Bannon area had completely collapsed by 1999, to where it sometimes would take me an hour just to find one plant with some mites on. And it was really frustrating. So we went out and we did a survey, and we found that there's this little guy right here called Phytocyalis persimilis. And it's a red predatory mite. And here you can see Gorse spider mite eggs. And when you would look in where the eggs were and where the nymphs were hatching, you'd find these guys. And they were just basically wolves running around eating all the baby sheep, the baby lambs being the baby spider mites. And they just wiped them out. If the Gorse spider mite had been a pest, this would have been an incredible biocontrol success. Only in this case it was not good because we had a biocontrol of a biocontrol. So this was in the coastal areas. The whole thing just collapsed in one summer. And then also at our inland sites where we thought, okay, we're going to be safe from there, we found these little tiny ladybird beetles called Staphylinus, and they're in there feeding on the mites. And here's the larva. You know, they can eat like 100 mites a day, and you can tell whether or not you have a predator in your mite colonies, because remember that one picture where everything was just covered with webbing? That's when they're predator free. But when the predators are present on them, it looks a lot like this, where you can see there are some mites. If I hold it up to a dark area there, you can you see the webbing in here? Looks like cotton. But can you see the webbing only goes about halfway out to where the ends of the stems are? That tells me there's predators in here. Okay, especially if this was in the summer. In the winter, not so much, but probably in here. And so what you look for when you're going to check these to see if they're predators, you can get these things, and I'll show you here in a second when I get to it, if it isn't the next slide. So anyway, the air just got sucked out of our happiness balloon, and the whole thing fell apart. And so we realized that the Gorse spider mite was not going to be a panacea, so we knew we had to look for something else. Okay, so I worked with Dr. Paul Pratt from Oregon State University, and what we wanted to do was do some experiments to find out what could we do to undo this, you know. So we got some insecticides and we sprayed gorse along cranberry bogs. This is over off of Rose Road over there. And we sprayed this out in there, and it's toxic to the spider mite predator, but not to the spider mite. So we sprayed that out there, and sure enough, the mites exploded and looked like everything was going great. And then by the end of the summer, they caught up with them and took it out. But what we showed is that it was definitely the predators that were maintaining the mites at these low populations. Now, can you go run around all over through Bannon and spray toxic insecticides to release the Gorse spider mite? No, that's impractical. But at least we showed there was a direct correlation with that. And so if it was just in a little area, you might could do that, but probably wouldn't do much good. Sad. Okay, so enter new possibilities for new biocontrols. And so we're working with scientists in Hawaii and New Zealand, and they have some other biocontrol agents that we haven't introduced into Oregon yet. The first one right here is this little teeny pot right here called the Gorse thrips. And thrips is for one. You have one thrips, you can have two thrips. So just so you understand. And let's see, this is what they look like. Here you can see them. You can see a lot of their feeding damage. They feed on the new growth. So this is something that we wanted to add as a, to build up our arsenal against gorse. And so here you can see the thrips right here, and here's a little bit bigger one. Really tiny. But when they hit these, the new growth, and we wanted something that would do that. And so the Gorse thrips has not yet been introduced into Oregon. We're working at it presently at the quarantine area. Now, because the Gorse thrips feeds on young succulent plants, we're trying to think of where would you utilize them, because in a lot of areas where there's old growth gorse, you don't see a lot of seedlings. So this is up at Baker Beach where they did this massive program, and they went in and sprayed and they bulldozed and then they burned. And these are those wetland areas that we heard about up in Washington, where you get a lot of the species, the riparian wetland species down in here, and just as soon as you get up above where the water kind of stays, then you get gorse. You get gorse, and this is just thousands and thousands of gorse seedlings right here that came in, and they had to be sprayed again. And this would be a place where you do this kind of restoration, maybe where we could get the gorse thrips in to help reduce the impact and survival of all those gorse seedlings in conjunction maybe with herbicides and things like that. Okay, in order to get the gorse thrips and another moth that we're looking at called Agonopterix ulicitello, which is a gorse soft shoot tip moth, we are studying them in the quarantine facility at Oregon State University in the Forestry Sciences building there in I think it's Rich Hall. And what we're doing in here is we put the thrips in with different test plants to see if they'll survive, and whether they'll feed on the non-target plants or whether they'll lay eggs or go through a generation and things like that. So we found that when we fed the thrips on just gorse, this is gorse right here and this is the fraction of gorse seedlings surviving, that you can see that the seedlings dropped off to about 30% success or survival at the end of about almost 60 days here. This is the height that the gorse seedlings get when they have the thrips, and here you can see the gorse without thrips just kept on growing. And so you can see that the thrips has a pretty important role in hopefully helping to reduce the gorse population from recruitment from seeds. So we did some other experiments where we took the thrips in our no-choice testing. And this is the survival on gorse. We had a couple of other plants that they survived on for a while. They never reproduced and maintained a population. One of them was, I think it's a Vicia, if I recall. Or was it something? Yeah, it's Vicia tetrasperma. And so we don't know why the thrips is feeding on that plant. We got them to feed on it and go, and they survived for a while, and we did get some reproduction on there. But one of the things we need to do now is to finish doing the testing on there and find out if they can go completely from one generation to another generation to another generation on a non-target plant. And if they do that, then we can't even think of releasing the gorse thrips because we don't want it. And I don't remember if tetrasperma is native or if it's introduced. It's introduced. So that is a mitigating factor there, that if it's an introduced weed and they feed on it, then we might be able to get away with it. But the problem is then it opens up, well, if it feeds on this vetch here, what other native ones might it feed on? And so we have to test a whole bunch of those. Because what happens here is you might find something that is totally unrelated that falls within its ecological host range or a physiological host range. And so we have to find out what's going on there. So we're doing the research to continue. We're also working with our colleagues in Hawaii where they have all the different gorse natural enemies working as biocontrol agents. This is up on the Parker Ranch. Were you out in that area, Tom? Yeah, so you recognize that. This is Gorse Central. It's up about, what, 6,000 to 7,000 feet. And there's two seasons there. There's morning where it's nice, and then there's afternoon where it rains. And so the dry and rainy season occur each day. But it's really an amazing place. It's just not a place you would have expected to see gorse, and a real problem. And this is a really neat area because it's a productive area where they graze cattle. It's also where the acacia koa, the koa tree grows up in there. And with all this gorse and the fire hazards and whatnot, that could be a real problem. So anyway, I'm working with my colleagues. This is Fritzy Grefstad, who's at OSU doing the quarantine work on gorse. And gosh, I can't remember his name right now. I will when I don't have to. Anyway, it's my colleague from Hawaii. And they're using my patented meatball entomology technique here to find the gorse mites, the predators, and the thrips. And that's, you go over to the gorse, you cut off a piece of gorse, and then you whack it like this really hard on the hood of your truck. And the mites and stuff fall off, and then you watch right here and you'll see the thrips running around. And actually, we found out that while it was sitting there and we were talking, thrips were falling off. And so we found out that instead of whacking, which worked really well to check for gorse predators, if you just talk to the gorse, the carbon dioxide would panic the thrips and they would fall off the gorse. And so you could just breathe on plants and do your thrips survey and then go over and whack them on the hood. It's really kind of cool because the gorse spider mites, they kind of crawl around really slow, you know, because they're sheep. But the wolves, the predatory mites, are really fast, and they're going around, you know, they look like little things going around really fast, and they're about 4 or 5 times as fast as the gorse mites. So you just whack them on the hood, wait for a minute, it's warm, and then everybody starts moving around, and if they're moving really fast, you know you have predators present. So this is one of those meatball entomology techniques that I'm famous for, but it works pretty well. It's not very scientific, but it works. And so that's what we were doing. And that's how we were doing the survey and trying to find the gorse insects and what was going on there. Now, another thing that they released in Hawaii back in 1988, I believe it was, or 89, they released this little moth larva right here called Agonopterix ulicitello, which is the gorse shoot-tip moth. And they feed on the soft part of the gorse, of the big plants. So we were hoping, since the mite fed on the old stuff, we'd get something that would feed on the soft stuff as well. And they had cages where they put them in here, and we go in with a bee smoker, smoke the cage, and all the moths would panic thinking, oh, my house is on fire. And they'd fly out onto the edge of the cage, and then you could harvest them and collect them that way. And that was kind of a cool way of doing it. So we went back over just last September, and we were finding that the gorse shoot-tip moth had done a pretty good job of dispersing throughout the area in Hawaii, and then we got more larvae and adults to bring over to the quarantine, because we're also in the early stages of doing the quarantine studies with this insect. But right now, the problem is the larvae will feed on a number of lupins, which is a problem. But that's the larvae. But the adults never lay eggs on anything but gorse. So the adult moth is what makes the choice of where it lays its eggs, and they never laid on lupins at all. In fact, we have a moth that I'll show you that occurs here that attacks Scotch broom and gorse both, and I've got its larvae to feed on lupins, no problem, but I've yet to find a larva on a lupin in the wild, because the adult is what takes care of the host specificity. And even when they're growing together in the same plants, I've still yet to find the larvae feeding on the lupins. So it's within their physiological host range, but it's not in their ecological host range. So it's kind of an interesting thing, and is going to require some more work. Okay, one of the cool things that I found, instead of going out with a smoker and putting up a screen, is if you just busted the gorse plants apart, which is no small feat, as many of you know, you look in here where the old gorse pods are, and you find the moths were inside there, because they look, they're about the same size and almost the same color as the old gorse pods, and that's where they would go in winter. So you could just rip the plants open and look for these things, and there the adults would be. And so it was an easy way to just kind of catch the adults during a time where we weren't working on them, and they were quiet, and they just like to sit there and wait for the winter or the cold season to go away. So this allowed us to collect quite a few of those. This is the one that we're looking at in Oregon. This insect on Scotch broom feeds in the flower usually, and you'll see them when if you worked on Scotch broom and you're whacking the plants looking for Scotch broom seed beetles and weevils, you'll see these guys on them, and you can always tell the genus Agonopterix if you have a larva, you touch them and they just turn into complete spastics. They wiggle so fast that they'll just wriggle right off of your hand, just like that. It's amazing. You'd think they were on meth or something. So that's how you can tell that particular thing. But here's what you look for. And down in this area and further south, especially down in Curry County, you come out in the spring and you look at the gorse here and you see instead of having normal twigs come out and expanding and growing, the tips are all dead and they're brown. And all of these brown tips are from this particular caterpillar. They web all this stuff together and they feed on the new and developing leaves, and they have a little tube in there and they stay in there, and that's where they pupate. And then the moth comes out and then they fly around and then overwinter in the gorse or in your garage or someplace like that. So this insect is pretty much everywhere, and one of the reasons we're kind of hesitant to bring in the gorse shoot tip moth is this is its kissing cousin and it's already here. And so we don't know whether it would do any good, because if you look at these plants, almost every one of these twigs is hammered by this insect. But what it does is, when you can tell when you're driving through, because if gorse, kind of grows its normal way, these guys aren't there. But when you see gorse that's really compact and lots of twigs, you can tell that this insect is there, because then these die out and then they branch out more, and it makes a much more compact, dense plant. So that's pretty much what I have. I don't even know what time it is to watch. So I have like, what, 5, 10 minutes? How much time do...
So you have 5 minutes, but then we're going to roundtable discussion,
Okay.
so that works out.
So I'll pass this around. You can look at, well, except you don't have gloves. If you're tough, or I'll just leave this up here and you can come and look at it. That'll probably be easier. And any questions? I'm sorry I kind of rained on your parade, but you got to know what the reality is. Sometimes for some plants we have silver bullets that are going to work really well, and for others we don't. But a little bit other thing to add is that there are some biocontrol projects that when they first started them they thought were failures, and they were failures for 20 years, and then over time, with the changes in land management, the decrease in disturbance and things like that, the insects collectively over time were able to stress the plants enough to tip that balance to where the weeds just kind of disappeared and became a lot less in the environment, to where they changed anywhere from 2 to 3 or 4 orders of magnitude in change. And it took 30 plus years for that to happen. So that's why monitoring is really important, because in some of these areas, if we don't do biocontrol of gorse, nothing will change, nothing will happen unless we change your management practices that we're all here to learn about. But in those areas where you can't get into, where it's too expensive, or there are important areas where you can't go into because of their ecological importance and things like that, that's where biocontrol will work, and that's where we'll work with you to identify those areas. Because before we can introduce the gorse thrips, we have to have some areas where we can do some pre-release monitoring. So that's one of the reasons why I'm down here, is to find out if you have some sites that aren't slated for intensive control, that we can have the gorse there left alone for 5 or 10 years, if that's even possible. Then those are the kind of sites that we're looking for. So, any questions? You're overwhelmed, or I did a really dang good job. Okay, well, I'm done, so on with the...